Hi, I'm David Bickle. I'm the instructor for the Bioinformatics Concentration. The Bioinformatics Concentration consists of 3 courses. So the numbers are IAB 620, 621, 622, Introduction to Bioinformatics, Bioinformatics, and then Advanced Bioinformatics. And I will discuss the details of these courses. The general idea for bioinformatics is that we have large data sets coming from genome projects where we have DNA sequences of humans and other organisms that have generated lots of data, not only DNA sequence data, but also protein sequence data, gene expression data, and related data from proteomics, metabolomics, and lipidomics. So bioinformatics deals with the tools, the mathematics and software for analyzing these kinds of data sets. So why study bioinformatics? Bioinformatics is a very general area in terms of different industries. Includes not only healthcare companies like pharmaceutical companies, such as drug development companies, but also agricultural companies such as seed companies. So I worked for Pioneer, a seed company in Iowa, part of DuPont, and we did a lot of bioinformatics there. And so that's where I picked up the industry experience that informs my teaching. In North Carolina, jobs can be found in this area using Indeed.com, searching under bioinformatics, and these are the first job titles that popped up when I did the search. So bioinformatician / machine learning scientist, information scientist, bioinformatician. So when I worked in bioinformatics unit, my job title was research scientist. So the job title can vary from one company to another, and using the same bioinformatics skill set. The courses I teach use 2 textbooks that I've written. And I want to highlight those since there are advantages to having the textbook author being your instructor. So you may have read textbooks in the past where you're not sure what the author meant. You can always ask me, what did I mean when I wrote such and such? So the first textbook I want to summarize is Phylogenetic Trees and Molecular Evolution, Hands-on Introduction with Uncertainty Quantification Corrected. And this one jumpstarts readers with a visual introduction to the main ideas of molecular phylogenetics. So phylogenetics is a term meaning estimated trees of where we assume that different DNA sequences or protein sequences are coming from the same common ancestor. Then we relate these and have some hypothesis about how the current sequences that we have, how they would be related in an evolutionary tree. And this book provides competitive dice games to equip players with intuitive grasp of evolutionary models. So these concepts are potentially complicated, but they're simplified when we play the dice game. So I break it down to one step at a time, starting with the graphics and then going to the dice games so that the concepts of probability and how they relate to the analysis of DNA sequence data or protein sequence data becomes clearer. This book features hands-on exercises to foster and assess mastery of the material and to stimulate critical thinking. So the exercises in the book are normally completed by teams of students, and the exercises are sometimes the games, but also working out problems and then answering questions to assess comprehension of the material. And then some other questions that are more open-ended that will bring students to think through the issues on their own and using the skills learned in the course. The other book I want to highlight that I also use in the courses is Genomics, Data Analysis, False Discovery Rates, and Empirical Bayes Methods. So whereas the first book was developed specifically for a course here at UNCG, this book was developed in my teaching over several years at University of Ottawa. And this one also involves dice games, interactive software to teach some of the concepts, and examples focus on gene expression and genetic association data. The same statistical methods involving false discovery rates can also be used to analyze metabolomics data and proteomics data. So those also are treated in the book. And this book gives a gradual introduction to the mathematical equations. So there are the equations that don't require any high level of algebra, but can be daunting in terms of having different symbols for concepts that are at first unfamiliar to students. So I start out very simply and build up one step at a time so that the material can be grasped. And these are the kinds of mathematics that go behind the software. So students get an understanding not just how to run software, but also what the software is doing and how that is improving the data analysis. So I'd like to go into the descriptions of the 3 courses in the concentration that use the 2 textbooks. So the first course, Introduction to Bioinformatics, in this one there are models of DNA and protein evolution. So this is where we get to the estimation of the evolutionary trees of how sequences may be related to each other if they did share a common ancestor. And the methods will be covered to estimate those relationships from the DNA and protein sequences. And then the book, this course, using the first book I mentioned, also mentions how to, teaches how to get hypotheses about common ancestry and the evolutionary events. So you can't have an evolutionary tree without assuming common ancestry, which is a reliable assumption when we're dealing with the recent past, such as looking at different variants of the virus behind COVID. And we could also, if we make that assumption of common ancestry, extend it into more distant past. A lot of this course will cover the uncertainty involved as we go farther and farther into the past, and how to quantify that uncertainty, how to propagate it to the results, so that we're not just claiming that these trees are the truth, but these are estimates, and that each estimate has its own uncertainty, and I show how to quantify that uncertainty using probability. But again, the mathematical level coming into the course is just very basic algebra, and so the concepts are explained in a simple way, one step at a time. This course, Bioinformatics, this is the second course in the concentration, second of the 3 courses. This one involves biological databases, sequence alignment, which you have to have the sequences aligned, to be able to compare the sequences for making the evolutionary tree estimates that were in the first course. So in some ways this goes back to some of the details needed, the background computations needed for the methods of the first course. This second course also has ontologies, reproducible science, and the main focus of the course is the statistics and machine learning needed to analyze gene expression data. So the statistical methods and machine learning methods are general, so they apply not only to gene expression data but to other types of data found in bioinformatics applications and even more generally. And yet I focus on gene expression data because it's a concrete example, and by focusing on that one concrete example of gene expression, the methods can be learned in more depth. Now, this is the third course, Advanced Bioinformatics. In this course, students analyze genomics data across thousands of genes or DNA sites, and this course is using the second textbook I mentioned. The students also use statistical methods for analyzing proteomics, lipidomics, and metabolomics data sets. So that's all part of this third course. And then at the end of this course, as well as the other two courses, the students critique conclusions of scientific papers. So the methods are learned in enough depth so that students will be equipped to read the scientific papers critically, being able to not take everything just because it's published, but to be able to understand the conclusions of the authors and what's the basis for those conclusions in terms of the bioinformatics methods. What impact do the bioinformatics methods have on the conclusions? Are the conclusions really warranted? Are they expressed with too much certainty, where the authors may be suppressing some of the uncertainty unknowingly? And how can the tools of these courses be used to understand how certain we can be in the different conclusions coming from the data? Thank you.